Good morning, everyone. Thanks for coming. It's my great pleasure today to introduce Dr. Petros Giannikopoulos. He is board certified in anatomic, pediatric, and molecular pathology and is a fellow of the College of American Pathologists. Which
Recording in progress.
How I know him. He obtained his MD from Harvard Medical School, completed his pathology residency at the University of California, San Francisco, UCSF, and trained in pediatric and molecular pathology at Texas Children's Hospital and Harvard Medical School, respectively. After his training, he became an entrepreneur for a little while in the areas of health technology and clinical genomics before moving to his current role, which is really how I became involved with him at this current role. So currently he's a principal investigator at the Innovative Genomics Institute, where he focuses on development and deployment of laboratory developed molecular diagnostics to support the advancement of CRISPR-based genomic therapies. And so I got to know Petros on a CAP work group that was looking at gene editing and the impact that it might have in pathology. So he directs the interventional genomics units, clinical laboratory, and the next generation sequencing platform at the Innovative Genomics Institute. And he's also an assistant adjunct professor in the Department of Laboratory Medicine at UCSF. And today he'll be talking to us about some of the CRISPR-based gene editing and the role that pathologists will play in that field. So turn it over to you, Dr. Giannikopoulos, and thank you for coming today, especially since you're traveling in Singapore right now. So I really appreciate it.
Well, thank you very much. And can you hear me okay?
Yes, we can.
Oh, great. Well, it's thank you very much, Sophia, for the introduction and for the invitation. This is a real pleasure for me. I think it's always great to talk about my work with fellow pathologists. I'm kind of I'm the only pathologist at my current place of work, and so what I'm excited to share with everybody here is how our field can actually play and is playing a very important role in bringing this powerful new technology to patients. And so, as Sophia mentioned, prior to coming to the IGI, I was a health tech entrepreneur for about 10 years. And, I started my first company. I mean, the reason I became an entrepreneur is because I was interested in developing platforms that could help patients overcome barriers to access to high quality care. And so I came to the IGI because we're kind of in this really interesting moment in the history of medicine where, for the first time ever, because of CRISPR, we are technically able to get at the root cause of many devastating genetic disorders. And so, the real impetus was not just, the vision for CRISPR becoming kind of the standard of care for genetic disorders, but, I wanted to make sure that, we get this powerful technology to everybody. So anyway, the role of the topic of my talk today is the role of pathology in this exciting new era of CRISPR-based somatic cell gene editing. And the goal today is, the goal here today is to provide everybody with a kind of an overview. And this is the Institute and Jennifer, our founder, and to provide everybody with an overview of how pathology and specifically the clinical lab is integral to this new and rapidly evolving field branch of medicine. And so, and just given the nature of the work at the IGI, everything that I'm going to be talking about today is going to be in the context of Mendelian disorders, although, gene editing is also playing an important role in oncology treatments in the form of mainly CAR T cells. So the way I'd like to structure the talk is to first provide a brief overview of the therapeutic toolbox of CRISPR and go over the landscape of the various approaches that people are using CRISPR to edit somatic cells. And what I mean by somatic cell gene editing, we're not editing embryos right now. so that's an important distinction. And then I'd like to discuss how we at the IGI at least are thinking about how to use clinical diagnostics in kind of the three clinical stages of editing-based therapies: the pre-treatment setting, the intra-treatment setting, and then the post-treatment/follow-up setting. So before I kind of jump into the meat of the talk, I thought I'd start by providing a quick snapshot of the landscape of laboratory methods that we use in kind of in this space. And I'm not going to delve into the details of all the different platforms or methodologies, but I just wanted to make a point, which is really, if there's going to be one take-home message, it's that and this is something I know I'm preaching to the choir here, but that any useful diagnostic assay really has got to exhibit two broad characteristics. Right? One is that the technology's got to be robust, right? It's got to be reproducible, and it's got to be accurate. And the second part is that the significance of the results of that assay need to be known. And in other words, if the doctor orders a particular test and he doesn't know what to do with the results, then clinically speaking, you're kind of in no man's land. And I know this is something that everybody here is well aware, but it's one of the challenges in molecular diagnostics today, which is that our ability to generate data has really been outpacing our ability to interpret the results. And so this has been really particularly true following the introduction of next-gen sequencing. And now we're entering a world of real kind of turbocharged single-cell sequencing and multi-omics and stuff. And so we've got a little bit of a chicken or the egg problem, which is that you need to deploy new assays in order to learn what to do with the results, but so that you can better utilize them. And so you can't learn unless you use them, but it's hard to use them if you don't know what to do with the results. So I won't go into the technical details of all the various tools that we're using, but the way I'd like to think about the different platforms is kind of along two dimensions. So one dimension is what I call kind of the biological dimension. You can look at DNA, RNA, proteins, et cetera. And then within each of those dimensions, you can also think of the spectrum of data volumes that you can generate, where classic tools like PCR or Sanger sequencing give you very, very robust but limited snapshots of information. But as you get kind of a little bit more advanced, like in single cell sequencing, you're talking about a fire hose of information. We're actively trying to figure out what all of these things actually mean. And so I'll leave that there. And with all that said, I'd like to start by saying a few words about the toolbox of therapeutic CRISPR editing. And maybe I don't know kind of what the technical background of everybody here is, but the first CRISPR-Cas systems were basically engineered with a guide RNA that can specifically identify genomic location of interest. And then there's a nuclease. So there's kind of two components to like the classic CRISPR system. There's the guide RNA that localizes the Cas nuclease to a particular site in the genome. And then that Cas nuclease can generate a double-stranded break. And then this double-stranded break can be used to either stimulate homologous repair, where a donor sequence can be copied to repair a mutation, or non-homologous end joining can result in indels that can disrupt a particular gene. And since 2012, there's been a massive proliferation of variations on this kind of core technology these modifications have basically relied on altering the Cas proteins in various ways to add various functions to them. So, for example, you can inactivate one of the two nucleases that are within the Cas protein and add a deaminase domain, and that creates base editors. And there's adenosine base editors that can convert adenines to guanines, and there's cytosine base editors that convert cytosines to guanines. And this is a table from David Liu's, this is a review paper from David Liu's group at Harvard. David was basically developed base editors. And so with these tools in hand, you can introduce a number of various genomic perturbations, such as you can knock out a gene by, generating a non-homologous end joining based indel, so you can disrupt, an allele. You can knock in a gene, so you can replace, the cDNA of a gene. You can repair a point mutation and many other approaches. And now people are actually you can add methylation domain to the Cas protein, and that's how people are modulating the epigenome. And so it's kind of creating a whole new class of therapeutics. But when we think about therapeutic editing, I mean, a lot of attention when people read about CRISPR, a lot of the focus is on sort of like what's happening within an individual cell. But when we're talking about editing in the context of a genetic disorder, there's several other key dimensions that also need to be addressed in this sort of preclinical phase of development that I want to briefly touch on. So the first is that for a particular disease, there's a specific therapeutic window in which generating a somatic edit will yield a benefit. So, for example, some diseases are lethal in the first few years of life and require intervention very early in the patient's life, while other diseases such as Huntington's manifest later in life, so you can intervene a little bit later. The other dimension is which cells in which tissues need to be edited and at what rate. So this all depends on the pathophysiology of the disease. So, even though every cell theoretically has a particular pathogenic mutation, it only manifests in certain tissues. And so we need to know what type of cellular perturbation we need to generate and in which cells in order to achieve the desired outcome. So, for example, in sickle cell, which is an area that my lab works on, so the IGI is part of a clinical trial with UCSF treating sickle cell with CRISPR. There's multiple strategies in clinical development, such as you can actually induce fetal hemoglobin by disrupting the transcriptional repressor of fetal hemoglobin, BCL11A, or you can actually edit the point mutation that causes the most common type of sickle cell. So, with all that said, I'd like to talk about how we use molecular diagnostic assays in these kind of three settings that I find very helpful in kind of organizing our work: the pre-treatment setting, the intra-treatment setting, and the post-treatment setting. And the reason I think this is a helpful framework is because, the purpose of any assay, as I mentioned, is to generate information that can address key questions related to a patient's care. And in each of these clinical settings actually has slightly different clinical concerns. And so, and this is a paradigm that's been applied in the past, and I think is deployed every time that a new therapeutic modality emerges, from solid organ transplantation. Here's a brief timeline of cardiac transplantation and the role that diagnostics played in its success to targeted therapies like Gleevec, which basically is kind of the poster child of precision medicine. And so anytime there's a new therapeutic technology, there needs to be the parallel development of the diagnostic frameworks to help guide who gets it and to monitor how well it's performing. So pre-treatment. So in the pre-treatment setting, this is before a patient gets treated. The key issues in this setting are determining who's likely to benefit and determining who may experience an adverse outcome. So it's basically thinking about inclusion and exclusion. So the first step in determining if someone is eligible or ineligible for treatment usually means establishing if they harbor, a germline variant that is known with great certainty to be causing the phenotype of the patient. So whether somebody actually is harboring a pathogenic variant. And then the second is whether the pathogenic variant is actually addressable by an available therapy. Now, admittedly, today, there aren't very many therapies available, but over time, as the medicine cabinet starts to proliferate, there'll be, this will start becoming a little bit more complicated. And so, as anyone who's ever, and I'm sure many people here on the call have been involved in germline testing can attest, determining whether a variant or a subset of variants detected upon sequencing are pathogenic is not a trivial matter. And, once you've ruled out that a variant is pathogenic or likely pathogenic, you're left with oftentimes a very large number of variants, the significance of which are unknown, or these are the VUSs, the variants of unknown significance. And this challenge has really grown substantially in recent years as we've started to look at more and more genes, or looking at whole exomes or whole genomes. One way that we try to get around this, and these are some recent data showing how not only are the number of VUSs proliferating, but also as we look at more genes, the number of variants of unknown significance in databases such as ClinVar increase, but also the classification. It's a dynamic classification system. I mean, over time, variants that are thought to be pathogenic may be reclassified as unknowns, as unknown, and vice versa. And one of the ways that we get around this is by looking at large numbers of patients and trying to draw conclusions based on statistics. But where this gets very, very challenging is in very rare diseases where it can take years or decades sometimes to get enough observations to draw any conclusions. And and so the problem is that these types of studies can not only take a lot of time, they may not always provide clear answers. And so for very uncommon disorders, a single laboratory or even a single biobank may never observe enough of cases to be able to adjudicate the significance of a particular variant. This is just an example of a very rare disease that one of my colleagues works on. And so collaboration, databases, those are critically necessary. So the other key point that I wanted to make is that that the performance of a guide in gene editing can actually be affected by the surrounding polymorphisms, both the polymorphisms at the targeted site, and also a guide can also cut or modify other regions of the genomes based on differences, the presence or absence of polymorphisms at different sites. And this just shows you the PAM sequence, this NGG PAM, this NGG the NGG sequence is what we call the PAM sequence of the guide RNA. And as you can see, that polymorphisms actually have resulted in very wide distributions of frequencies of PAM sequences across the genome. And so what that means is that depending on the haplotype of an individual patient, you may see very different rates or different efficiencies of a guide at the on-target site, and also very large differences in off-target activity. And this is a very recent paper from Luca Pinello and Dan Bauer that showed that how the presence of a polymorphism at a known off-target site for a guide RNA that targets the BCL11A gene, and it's a sickle cell guide RNA. You can see here, this shows you the in silico analysis of the off targets for a particular guide. And here, what it's showing you is that the presence, so this is the off target profile of a guide using two different Cas proteins. And when compared to the reference sequence, you can see here there's actually a much higher rate of off target involvement. So when we're evaluating the on-target, when you're assessing the on and off target efficiency of a particular guide, the patient's genome, the sequence matters quite a bit. So, and finally, the other thing, and when evaluating whether or not a patient is likely to benefit or likely to experience an adverse event, it's also important to know whether a patient is at risk at baseline for developing things like leukemia. So we know for sickle cell disease, it's known that myeloid leukemias can emerge after autologous gene therapies, and gene editing therapies that utilize myeloablative conditioning can also result in leukemias. So some clinical trials are choosing to exclude any participants that have evidence of like myelodysplastic syndrome or clonal hematopoiesis. And we know that for sickle cell, it's been shown that that patients, gene editing aside, are at higher risk of developing leukemia. And recent studies like this one here, a couple of years ago, have shown that that in sickle cell, you see the evidence of earlier and more frequent clonal hematopoiesis, and this is also true in other inherited hemoglobinopathies, and other inherited hematologic disorders such as congenital neutropenia and bone marrow failure syndromes, and I think like Shwachman-Diamond syndrome. And the challenge, of course, is knowing, what we don't right now quite know what the long-term significance of some of these mutations that we identify. And so, but this is one very important role for molecular diagnostics in this pre-treatment setting. So now turning to intra-treatment assessment, it's important to start by distinguishing between ex vivo and in vivo editing. And those are two very different approaches. So ex vivo editing is by far right now the most common form of therapeutic editing in the clinic. And it consists of essentially a three-step process. So step one is mobilizing and collecting cells that are to be edited. And these are typically either T cells or hematopoietic stem cells. And then step two is editing the cells using one of the various strategies that I mentioned previously. And then the third step is transplanting those cells back to the patient. So in the clinical trial that my lab's a part of at UCSF, which is being led by Mark Walters and Don Cohen down at UCLA, the bone marrow derived stem cells are harvested. The sickle cell point mutation is corrected using an HDR based approach. And then the stem cells are then transplanted back into the patient. And now in vivo editing, this consists of an intravenous infusion of either viral particles, viruses, or viral-like lipid nanoparticles that deliver the editing payload to the desired cells. And this is typically the liver, because you can actually get target the liver using things like AAVs. And but for the purposes of our talk today, I'll focus mainly on the ex vivo edited drug products, since that's right now what most of the trials are. But you're going to be seeing a large proliferation of these in vivo trials that target not only the liver, but also the central nervous system, and also just even in vivo targeting of bone marrow and even immune cells. So when we actually assess the quality of an edited drug product, there's basically there are some characteristics of the drug product that are independent of what gene is being targeted, and that's also resolvable quite readily with clinical diagnostic testing. So for cell products, one of the most important of these characteristics is sterility testing, which is to ensure that the product is free of any microbial contamination. And every IND sterility testing method are part of all the what we call kind of a CMC data, and at a bare minimum, this is something that this is sort of like a must-have for any drug product. And so one of the most important is testing for mycoplasma. And so I won't get into the details of how kind of this is done, but I just wanted to make the point that the way that sterility testing is done was really kind of designed for large scale pharmaceutical industry batches where you basically, synthesize a giant vat of drugs. And so what's happening nowadays is that people are developing more sophisticated approaches, more, molecular-based approaches for looking for microbes. And so here's just a recent example of like a group that's trying to do multiplexed microbe testing in drug products. But for the FDA still relies on some of these kind of big batch, old school methods for evaluating drug products. Now, when it comes to looking at the on-target specific characteristics, a common way to organize these characteristics is to separate those related to the intended perturbation, and that's what on-target activity or the term on-target refers to, and from the unintended effects, so which are what we mean by off-target effects. And so, another key point, as I mentioned before, for lab diagnostics in general, is that there's kind of two inherent challenges when we use molecular diagnostic tools when we're assessing kind of on and off target effects. One's the technical challenge of measuring whether something, whether a particular molecular feature is present, and the second is knowing what to do with those results. And so a lot of what I'm going to discuss has been published by the FDA. Put out a human gene therapy products incorporating human genome editing. This is one of the reports that the guidances that the FDA puts out, and so that if anybody's interested, they can kind of take a look at that. But when you think about on-target editing efficiency, it's important to kind of come back to this framework that I introduced before. And so for each disease, there needs to be some empirically based threshold for, there needs to be some, the standard, you need to basically establish, standards for each of these dimensions. So for example, in the sickle cell trial, it's known that based on many years of clinical data from bone marrow transplant recipients, that an editing threshold of approximately 20% of HSPCs is going to give you a sufficient clinical benefit. And so for every disease, the target of the efficiency or the threshold for what is satisfactory is very much disease specific. Now, for the current state of the art for how to measure on-target editing efficiency, so this is looking at the level of the genome, it's to use next-gen sequencing to sequence the PCR amplicons of the target site. And so if one is using an HDR-based approach, which is what we're using in our sickle cell trial, what you'll find when you analyze the data is you'll get three types of alleles. There's those that are unchanged. So you'll get the PCR product. When you sequence the PCR product, you'll get unchanged alleles. Then you'll get alleles that have indels, which are due to the non-homologous end joining. And then you'll get the alleles that have the desired edit. And so, I don't have here, I'm missing my slide here, but I was going to show you a slide of what the raw output looks like. But one of the problems when you do these types of bulk sequencing is that what you're getting is an average of all the cells in a population. So we can't tell the difference between 20% of the cells in a population being homozygous for an edit, or if 40% of the cells in a population are heterozygous for an edit. So although we're not talking about like in the case of lentiviral-based gene editing or gene therapies, people oftentimes use a statistic for the LVIs, or the lentiviral insertion. So they'll actually measure the number of lentiviral and there's an estimate for the total number of lentiviral inserts in a whole population, and then you'll get a number like 1.7. So like, but there's no such thing as like 1.7 lentiviral inserts in a cell. So that's just an average across a population. So one thing that's actually been very, very powerful over the past couple of years is single cell DNA sequencing. And so single cell DNA sequencing has started to emerge as a tool for generating more accurate on-target editing efficiency. And so one of the leading platforms for doing this type of work utilizes droplet microfluidics to generate barcoded PCR amplicons from tens of thousands of cells. And that basically enables us to determine the on-target activity within an individual cell. And I'll come back to this important point when we talk about off-target editing. The other key on-target characteristic that's really important for evaluating a drug product is the functional impact of an engineered edit. And so, as everyone here is well aware, these, or may be aware, these are called potency assays. And so, the term potency assay kind of came out of the gene therapy world, and these refer to laboratory tests that measure the biological activity or characterize the biological impact of a therapeutic, of a gene therapy. And they're really an essential part of determining whether a drug product is adequate or not. And for gene replacement therapies, like old school gene therapies, like inborn errors, like if you're replacing an enzyme for like inborn errors of metabolism, the strategy for designing a potency assay is fairly straightforward, since you're basically restoring some function that's missing. But for other conditions, it's a little more challenging. So for sickle cell disease, just to keep coming back to sickle cell, even though the DNA sequencing methods I was just talking about can tell us the extent to which alleles in a population of HSPCs have been edited, conventional hematologic assays such as HPLC, they can tell you whether the abundance of different hemoglobin variants in a sample, but there's no right now consensus approach to measuring the percentage of HSPCs in a population that have had restored normal function. Because the challenge with sickle cell is that what you're fixing is the propensity of a cell to sickle in some type of in vivo environment. And so this has been a real this has been a real challenge for the field right now, because all the assays that look at, there are commercial tools that can look at, sickling, but those have been optimized for blood that comes from a patient. So what we're talking about here is if you take HSPCs that get edited in a dish, and then you differentiate them and then what you get is a lot of artifacts that, and basically in vitro differentiated RBCs do not look like or behave like in vivo differentiated RBCs. And so, so this is an ongoing challenge in the field. So, so this here, I just showed, this is an example of, we're right now working on a method for characterizing the potency of a sickle cell therapy using a battery of different assays, and then generating a composite score that will tell us what percentage of cells in a population will actually, have restored wild type phenotype. So I can talk a little bit more about this, but it's just the punchline here is that this is an unsolved problem right now for sickle cell disease, but it's a critical piece of every gene editing program. So I'd like to now talk a little bit about what's really probably the most hot button issue in the field, which is the off-target effects of CRISPR editing based therapies, which when compared to prior forms of gene therapies, they are less worrisome, I should say, because CRISPR in theory is a lot more specific, but they're still present, meaning that even though CRISPR is a lot more specific, it has not eliminated the off-target consequences. So, a key reason we care so much about the specificity of editing is because, the risk of oncogenesis that can result from disrupting tumor suppressors or the activation of a proto-oncogene. And so we're reminded of this recently when, high-profile iatrogenic event was reported in a lentiviral-based sickle cell trial. And, the patient presented with AML a few years after the dosing. And, and although limited public information is available, the cancer was, I believe, described as carrying driver mutations very common in AML, and it wasn't due to the therapy, but for investigators and drug developers working on ex vivo editing of HSPCs, these iatrogenic events were reminders that we need to rigorously monitor for the possibility of, increased risk of malignancies. And then the other recent events that have raised the significance of monitoring for off-target effects has been several high-profile papers that show that Cas9-mediated editing can result in rare events of chromosomal arm level copy number changes and even chromothripsis. And these are some of the papers here that also and these are pretty these events, for example, I think it was like 2 or 3%. This is a paper from David Pullman and Mitch Weiss, showed that you can see in a population of HSPCs, you can see in the sort of low single-digit percentage evidence of chromothripsis, and more recently, and this is from Gang Bao's lab at Rice University, you can also see kilobase scale insertions and deletions near a gene editing site. So this was actually using a guide RNA targeting sickle cell, the HBB, the hemoglobin beta. And so the key question at this time, however, is that we just don't know what the functional and potential pathogenic consequences of these events are. And the worry is that, like for lentiviral therapies, that these rare chromosomal changes can result in cancer down the road. But there's also concern that maybe these off-target effects, these chromosomal changes, can also affect the differentiation of stem cells, which can result in cytopenias and other, functional consequences. So I would say that another kind of take-home point of all of this is that, we need a balanced and, I would say, committed approach to collecting preclinical data and long-term outcome data from patients over time in order to understand the significance. So, even if, just detecting these events is not enough, we also need to know what they mean. Another important challenge that's emerged recently, and this is a much bigger issue for in vivo editing based therapies, is, and these are for, diseases like muscular dystrophy and retinal disorders, is the activation and engagement of the patient's immune system. Now, and this is because, both, in vivo based therapies expose the host, the patient, to novel materials, and now those novel proteins carry the potential to activate the immune system. So, we must not forget that the CRISPR tools, the editing technologies, they rely on components that are derived from microbes. And those microbial proteins can trigger the innate and adaptive immune system. And this also includes, by the way, the viral vectors that we use to deliver the editing machinery into host cells. Now, in ex vivo editing, you can get around this by using electroporation or other non-viral mediated delivery. But for in vivo work, you can't get around the exposure of cells to bacterial protein, since the Cas protein is the Cas nuclease itself originates from bacteria. And so this is a paper from Matt Porteous's group at Stanford, and they've shown that in patients and in preclinical animal models, you can see varying levels of antibodies against Cas9, and in the mice you can also see activation of innate and adaptive immune responses against the protein itself, against the Cas protein, and also immune reactions of the edited cells themselves. And so this, of course, is really worrisome, because this limits the efficacy of the therapy, and could actually limit could lead to inflammatory side effects in the patient. And so there's also some recent papers looking at in Duchenne's muscular dystrophy based therapies. There was some these autoimmune reactions actually lead to a abrogation of the benefit. So in many ways, I think a lot of what I just talked about with respect to inter-treatment assessment is, it's part of the preclinical process, and it's also deeply woven into the IND enabling studies. So, in other words, it's a little bit like using surgery as an analogy. A lot of these tools, like the inter-treatment assessments, a little bit like, for those of you who do like frozen sections, or during surgery when you do like a CT scan or an MRI during the surgery to help make sure that the procedure is going according to plan. But when you look at the months and years following following a treatment, in the years following treatment, the use of molecular diagnostics and the role of the clinical laboratory starts looking very familiar. So I had here we go. I forgot my slide. So so what we're looking for in the long term, after in the post-treatment setting, is the durability of the genomic modification, and like the meaning, like, you want to see that the genomic edit is persistent, and you also want to make sure that that the functional benefit is also maintained over time. So, for example, in the CRISPR-based trial for sickle cell taking place at UCSF, we're using next-gen sequencing to look at the persistence of the edited allele in peripheral blood and bone marrow samples. And as we described right now, in conjunction with that, even though there's no ideal, there's no perfect potency assay, what we would be doing is, we'd be also looking at the sickling rates. That would be an example of like a long-term potency assay. So I think the last thing I wanted to, I had these slides were a little bit out of order here, but I think like what I wanted to end here is I think the question that's on a lot of people's minds, and this is the kind of last thing I'll touch on, is what should, what are the assays that we should be running, or like what's the what's sort of like the must, what are what's the playbook for what we should be doing? And so, and here's where I think, I wish I had more definitive things to say. But I think the way I like to think about this is that this is the framework that we've at the IGI has been using to think about the challenge of using molecular diagnostics in the context of these therapies, is that we need to create basically a learning system where we need to where we need to proactively deploy the technologies so that we can create knowledge about what's relevant. And once we know and this is a little bit of this is a grid here that we've developed that, the way you can think about molecular diagnostics in the context of CRISPR is that, you either know what needs to be measured or you don't, and then you have and then there's the capability of being able to measure what needs to be measured. So if you have knowledge of what needs to be measured, and you have the technology to and the technology to measure it exists, then that's kind of a no-brainer. If we don't have the technology to measure something, but we know that we need to measure it, then we need better technology. Sometimes though, and this is what I think really where we are right now in a lot of cases, is that we have the technology to measure certain things like the chromothripsis, like chromosomal copy number changes, but we don't really know how what that means. So and I'll just end here because I want to leave enough time for questions. And so a couple of take-home points. I think the first is that that success in the clinical lab requires both technological innovation and the clinical insight into what's important. And so these go hand in hand. I think the bottleneck ultimately, though, is knowing what to look for and why. And so, technological innovation, I think, always precedes the clinical insights. So it's just, I think one of the challenges is simply narrowing the gap between when you develop the technology to be able to measure something and then knowing what to do with that data. Molecular diagnostics are necessary for evaluating patients prior to them
Recording stopped.
Okay. Prior to them being treated. And they're critically important for predicting who may experience adverse effects. And in the context of somatic cell gene editing, the clinical lab plays a critical role for determining the quality of a therapy. Off-target activity is a major and constantly evolving challenge. Right now, I can say that, unfortunately, the FDA, and I'm part of the somatic cell gene editing consortium. And right now, everybody's kind of looking at each other, trying to figure out what we should do for a lot of these off-target effects. Groups right now are basically kind of focused on using base editors. These are the editing-based methods that don't introduce double-strand breaks in the genome. And obviously, it's going to be critical to monitor patients over time, long term, and that will be critically important for determining what's important to look at. What will be important to look for in the early. There's sort of like the long-term monitoring will be important, learning and a feed to inform kind of what we look for early on. So I'll stop there. And I can double click on any of these areas, but hopefully that was a good enough primer. And I apologize if that was either overly too simplistic and too high level, or too kind of off topic, but I'm happy to answer any questions.
Thank you. No, I think that was good. We have a wide range of attendees. So, and I think our recording stopped because our recorder, Amy Olson, must have lost her connection. So because I don't see her on right now. So I'd like to open it up for questions. If you want to unmute yourself and ask questions, or type something in the chat, I can ask the question. Hey, Leo.
Yeah, hi, Sophia. Petros, thank you very much. That was a absolutely fascinating talk, and thanks for taking up your evening in Singapore to meet with us. It's most appreciated. So I just have a question for going forward. We know from bone marrow transplants that even using, and stem cells, we know even using a hematopoietic cell population, in those cells are pluripotent stem cells, because you can find donor cells in all sorts of organs. And I wonder to what degree that presents an opportunity for thinking about various diseases or a problem going forward with bloods.
Well, when depending on the protocol, I know that for when doing ex vivo editing for hematopoietic disorders, or even for certain immune disorders, we do select for the stem cells. So we actually select for CD34 positive precursors, and so that is so we do, you know, so we do try to select for the stem cells in order to ensure that we are editing, you know, the cells that we're targeting are early. One of the big unanswered questions, and it's unclear, is to what extent, like because when you edit a population of stem cells, it's an imperfect, meaning that you don't actually edit all of them, and you get varying degrees of success. And there's various ways to edit them. You can use electroporation, which is, you know, zapping the cells, and then, you know, that they take up DNA and protein, or you can use viral particles and so forth. And so all of those different modalities can impact the degree to which those stem cells engraft, and then how they survive. And so it's you know, and so the way that people do it right now in the preclinical setting is you use mice and look for kind of long-term engraftment, but we don't really know. And this is a big part of the preclinical process, is trying to evaluate you're trying to correlate the efficiency of editing with the long-term outcome. But can you maybe kind of like, I'd love to know a little bit more about, like, specifically, like maybe clarify a little bit, like about your question. What aspect of the stem cells are you?
Well, in the population of what we call hematopoietic stem cells,
yeah,
are cells that are actually pluripotent.
Oh, yeah. Okay.
And they can, you know, you can find cells in the liver, in the brain, all sorts of other things.
Yeah.
That actually was observed many years ago in the early stages of bone marrow transplant, actually in Minnesota, where the first successful one was done. So it's, you know, it can be used as a tool if you've got, other than hematopoietic.
Oh, I see.
Something in the skin, or
yes. Oh, I see. Okay. I miss. Yeah.
I'm sorry. I wasn't more clear.
I missed that part. Yeah. So there's actually been an effort to basically either create, you know, induce pluripotent stem cells. You can make iPSCs, and one of the PIs at the IGI, Dirk Hockenmeyer, who runs the Stem Cell Institute at Berkeley, his lab is very, very good at editing iPSCs, and the hope was that you could actually edit the iPSCs, the pluripotent stem cells, and then differentiate them into various cells of interest. And the challenge there is, just in talking, I don't do this in my lab, is that it's really, really time consuming and also inefficient, but we're getting there. So there's, you know, some cell types are easier to generate in vitro. But the hope was that, and I think Stu Orkin's group at Harvard was doing this, where you could take the iPSCs and then, turn them into T cells or B cells or NK cells and so forth. And so it's some people have been talking about trying to create like sort of a bone marrow on a chip, different approaches for trying to edit iPSCs and then getting them to differentiate a little bit more efficiently. So yeah, it's a huge area of research right now, and I think there's a lot of opportunity. If you can edit the iPSCs, then theoretically you should be able to differentiate them into a cell of interest and then transplant those. Yeah.
Thanks very much.
Sorry for it's a little late in my process.
Oh no, not to worry. No, you did great.
All right. If no one else has a question, I have a question for you, Petros. How long do you think it will be before, less specialized pathology labs that aren't doing this in the setting of research are going to be? I mean, how long do you think it'll be before this therapeutic approach is a little more widespread?
Well,
I know that's future predicting, but
well, there's two things in terms of I know that we hope that both the cost and the cost will hopefully come down over time, and then hopefully the level of technical know-how necessary to produce and deliver the therapies will also hopefully come down over time. I think for the in vivo, so for ex vivo editing, I think that I don't know exactly when, but I think the bone marrow transplantation is probably a good sort of model where 40 years ago it was very, very few places that I think were doing BMTs, and now it's a little bit more common thanks to technology and sort of a lot of know-how. There are technological innovations that will, I think, make it a little easier to produce more, to be able to do this. Specifically, if you can do things in what we call kind of a closed system, where you don't need a large GMP type of facility, that will help out quite a bit. The other hope, though, is that you can, even for what's right now for the hemoglobinopathies and inborn errors of immunity, if you can actually just deliver the editing payload intravenously and basically do things in vivo, that hopefully may also make things a little easier to administer. But I don't think it'll be I don't think it'll be too far off before. I think the biggest challenge, so there's basically, in my mind, there's sort of two big challenges. One is delivery, because there's many cells where we just right now, we just cannot deliver the editing payloads very efficiently. I'm thinking about things like the heart, even the lungs. You can get them into cells, but where either there's low levels of efficiency or there's not sufficient levels of specificity. That's one thing. And then the other challenge, so delivery is one challenge. And then the other is, I think, just knowing what to edit. And this is something that I've been thinking a lot about. I mean, I think technically we'll be able to just about fix anything. But then for most patients with genetic disorders, we just won't know what to do, because, you'll sequence a patient with a very, very severe genetic phenotype. You do the whole genome, and you either A, don't know what's pathogenic or not. And then even if you do, it's very important to understand the mechanism of pathogenesis, because it's very difficult to craft a therapeutic plan without knowing what's going on. So I think it's going to be very important. I think we'll probably see, we need to learn more about specific diseases to actually know what to do therapeutically. And I think we just need better delivery technologies and tools. And we're seeing both right now. I think the brain, CNS delivery, there's lots of progress happening there. Liver, of course, is there, but that's a lot of the interesting stuff. And people are doing really exciting things like viral-like particles where you can actually conjugate antibodies on the surfaces of like lipid nanoparticles to get the lipid nanoparticles to home to certain tissues. And there's a lot of stuff kind of coming down the pike there. And the third, I would say, other big barrier is like the regulatory framework. I think the vision, the hope with CRISPR was that it'd be a little bit like the ASOs, the antisense oligonucleotides, that these can be a programmable therapeutic. The FDA right now still views, even if you change the guide but keep everything else the same, they still consider that to be a different therapy. And so you've got to go through the whole GMP rigmarole. So that will need to change if this is going to scale. So those are kind of, in my mind, the three big challenges.
Okay, well, thank you, Dr. Giannikopoulos. I was doing good until now, Petros.
I'm Petros. Yeah.
Thank you so much. We have a meeting at 9, so we need to end it here. And but I really appreciate you spending time with us.
Thank you. It was a real honor and really appreciate it.
Yeah. Just if anybody left a cell phone, it's in Amy Olson's office here beside me. If anybody left a cell phone, it was there yesterday after CP conference. Thanks.
Right.
Okay.
Right. Bye-bye. Thank you.
Bye.